We'll hear from Gauri Yogalingam, a scientist who recently joined the Cure GM1 board, who knows the beta-gal enzyme inside and out, as far as I can tell, and has about 30 years experience in ERT for lysosomal storage disorders. So,
hi everyone, I'm Gauri. I just became a board member of Cure GM1, and I'm going to talk today about ERT, but the lady at the back there gave a great talk about the importance of DNA editing for our own children someday, which would be ideally the best cure out there. But in the meantime, enzyme replacement therapy is something that's clinically available for a lot of other lysosomal storage diseases. So we thought it would be good to go through the work that we're doing to fund a Cure GM1 Foundation-led ERT, and for me to give you a bit of an overview on lysosomes and just how incredibly wonderful I think they are scientifically, and also how we can develop drugs that target the lysosomal system for GM1 kids. So if we take a look at a cell and we do a cross-section of it, the biggest organelle in a cell is the nucleus, which has our genetic material in there. And then in between the nucleus and the periphery of the cell, you have a soup. And inside that soup, which is called the cytosol, you have a lot of different organelles floating around in there. And one of them is a lysosome, which is highlighted there. And the lysosome is just a small little sac. It's an acidified sac, and it contains lots of different lysosomal enzymes. And there's actually hundreds of these lysosomes floating around in a single cell in your body. And they play a really important role in removing cellular waste. All day they're roaming around the cell and they're clearing out the trash that accumulates, because during a whole day of metabolism, the cell's creating a lot of cellular trash and it needs to get disposed of, and that's what lysosomes are there for. And this diagram sort of nicely exemplifies the importance of lysosomes. I think Christine showed a dirty bathwater example, and I'm showing you a dirty kitchen here. So if your lysosomes are working well, then you're going to have a nice clean kitchen here. There'll be work surfaces where you can cook your cut up your veggies. You're going to throw out the trash at the end of the day. You have a clean kitchen or a clean lysosome. In a lysosomal storage disease, you're missing an enzyme which leads to storage of material, cellular waste. And so in this kitchen, nothing's being thrown away. Everything is being hoarded in the kitchen and it's no longer functional. And so that's what happens in a lysosomal storage disease. So what does that look like under the microscope? With an electron microscope on the left, you can see a skin cell from a healthy donor, and you can see that their cell looks nice and clean. But on the right-hand side, this is a skin cell from an infantile GM1 patient. And this baby's cell is filled with these little bubbles, and each one of those represents a lysosome that's filled with lysosomal storage material. It can't get rid of it. The cellular trash is just accumulating, and those lysosomes are getting bigger and bigger to the point that they eventually bust open the lysosomal membrane, and they end up causing the cell to no longer be able to function. So what do lysosomes look at the light microscope level? This is about, I'd say, 20 times the magnification, 20 times magnification. And you can see that lysosomes are really cool. They're just moving around the cell continually. This is a cell from a lung cell, and each one of those little dots corresponds to lysosomes, and they're constantly moving around. So then if you go to the next slide here, that same video, if you take an image of that cell at 0 minutes, 1 minute, and 2 minutes, and then do an overlay in green, blue, and red, you get a cell that's full of sprinkles, and each sprinkle represents a lysosome that's been moving. So it just gives you an idea of how important it is for lysosomes to be able to move. In addition to be able to clear cellular waste, they have to be able to move around the cell to clear that cellular waste. So just a little bit of a history on the side. The lysosome was discovered in 1955 by accident. Christian de Duve was a scientist, and he was doing experiments in the lab, And he was really interested in looking at how glucose is made by your own body through a process called gluconeogenesis. And he discovered lysosomes by mistake. He found that he decided to have a long lunch break, and a bit longer than usual. And what he found was when he came back, there was now another enzyme that he could detect in the liver homogenate, and it was all of a sudden there, but it wasn't there before his lunch break. And also this liver enzyme could only be detected at under acidic conditions. So he hypothesized that maybe lysosomes exist, that you have these little membrane sacs that are containing lysosomal enzymes in an acidified environment and protecting them from the rest of the cell, and maybe they play a role in digesting cellular waste. And so he came up with this idea, he went on to prove it, and he won a Nobel Prize. But what it shows is that we should always be willing to stop what we're doing and move in a new direction, should it come along. So even if ERT works tomorrow, if all of a sudden gene editing works, we should stop what we're doing now and get that to work. So you should always be willing to move. And so he did. He stopped working on glucose, metabolism and synthesis, and he moved into the field of lysosomes. So what's been done since Christian de Duve discovered lysosomes 70 years ago? The lysosome is now 70 years old this year, 2025. 70 different lysosomal enzymes have been identified that are inside these little acidified sacs in our cells. Each one of those lysosomal enzymes, if it results in a deficiency in a patient because the mum and the dad have passed on genes in mutations that will result in mutations in that lysosomal enzyme gene, if that enzyme is deficient, you get a lysosomal storage disease. And all of the enzymes are different. They all play essential roles in clearing out different types of cellular trash. It could be lipids in GM1 gangliosidosis. It could be specialized types of sugar in MPS or mucopolysaccharidoses. So I've just highlighted two of the enzymes here, beta-galactosidase, which is deficient in our GM1 kids, and aryl sulfatase B, which is deficient in MPS children. And I'm just going to talk about how we developed enzyme replacement therapy for kids with MPS 6 or mucopolysaccharidosis type 6. And in this disease, they're missing the lysosomal enzyme aryl sulfatase B, and it's got a frequency of about 1 in 200,000 patients, so it's a rare disease. And if you look at using an electron microscope at the patient's cells, their lysosomal storage disease isn't occurring mostly in the brain, it's occurring in the joints, and it results in very short stature. And if you look at the lysosomes, they're in the chondrocytes, a type of cell in the bone growth plate. It's filled with lysosomal storage material, and because of that, the bones can't grow properly. So that's one of the major symptoms that MPS 6 children have. And on the right-hand side, you can see that looks like a healthy cell, and you can barely even see the lysosomes because the lysosomes are working. So it just happens that with MPS 6, just so in the same way it occurs in patients, it can also occur in cats. A lot of people like to breed cats, and there's a Siamese cat population out there. They seem to have a very high frequency of 2 different MPS mutations. And so it results in Siamese cats that have been inbred and ending up having either severe MPS 6, where they have a lot of joint problems and they're very sick. They have hepatosplenomegaly, and the milder version, the cats have very, very mild disease, where the only symptom that they have very, very late in life is degenerative joint disease. So there's like a really big difference there between a severe cat and a mild cat. And really surprisingly, the severe cat has no enzyme activity, but that mild MPS 6 cat only has 5% of enzyme activity. So the big learning lesson that we had in Australia when we were developing this enzyme therapy for MPS kids is that you don't need to get 100% of enzyme activity. You just need a small increase, as little as 5% increase in your enzyme activity in the patient's cells is all you need to go from having a severe lysosomal storage disease to almost being as good as normal. So we took advantage of this, and another thing that we found out along the way is that lysosomal enzymes have this really cool characteristic where, in addition to going to the lysosome when they're newly synthesized in a cell, they can also be released extracellularly. And they have a special tag on them, a mannose residue, a type of sugar, that acts as a beacon. And so once they're released extracellularly, they can be taken up by another cell. 1 more minute? Oh, 5 more minutes. Oh, cool. They can be taken up by a neighboring cell and targeted to the lysosome of that neighboring cell. And so that's something we can take advantage of with enzyme replacement therapy. So what we do is we take a donor cell from a healthy person and we start to overexpress the gene of interest. And actually the donor cell here isn't even a human cell, it's a specialized secretory cell from a hamster ovary of all things. And it just happens to be a really good secretory cell, and if we overexpress our lysosomal gene of interest, the enzyme gets secreted in large amounts, that healthy, good copy of the enzyme. And then we grow these cells for 14 days in a special environment with lots of special ingredients to make them really spit out that healthy enzyme. And then we pass that medium, that extracellular fluid, over a series of columns, and at that point we're left with this purified enzyme. And what we did, I think it was around 25, 30 years ago now, is we put that enzyme into the MPS 6 cats, the severely affected cats. And what we showed is that those severely affected cats ended up having a very, very mild form of MPS 6. For the most part, they were pretty much normal. So that sort of proved the power of ERT. And when we looked at the cells under a microscope, the severely affected cats, they had lots of lysosomal storage in their bone cells, but following enzyme replacement therapy, they looked indistinguishable from the normal cats. And they actually grew normally. If we gave the cats the enzyme replacement therapy when they were kittens, they grew up to be normal cats that were running around, whereas the untreated cats couldn't walk. They had hind limb paralysis. Oh, and I guess the other point here is just to say, back in 2005, this enzyme replacement therapy program got out-licensed from my lab in Australia, where I was working, and it got taken up by this biotech company, BioMarin, based in the Bay Area, and it got developed and approved for enzyme therapy for patients with MPS 6. And today it's been transformational for those kids that have MPS 6, and it's commercially available. So we're now trying to do the same thing with beta-gal, and when I was working at that company, it's BioMarin, I was very fortunate to lead a program to do beta-gal enzyme replacement therapy for GM1 patients. And unfortunately the program got terminated along with some other ERT programs, but luckily we got to do the experiments, and we showed that we can make beta-gal enzyme just like I showed you. We can make it to scale, we can purify it. When you incubate an infantile GM1 patient cell line with this enzyme, the purified beta-gal can now be detected in green in this image, and it co-localizes with lysosomes, which is shown in red, and green plus red equals yellow, so you can see nice co-localization. That means that your enzyme is getting delivered to the lysosomal compartment. That was in a patient cell, and if you look at that patient cell using another marker, GM1 gangliosides, the lipid that's causing the disease, it's a disease driver in these patient cells, that ganglioside is really present in quite a large amount in this patient cell line. And then after being incubated with just a tiny little bit of this purified beta-gal, after 18 hours, the majority of the GM1 ganglioside has been degraded. And if we wait longer, it's completely gone, and it remains gone for several weeks, just that one little dose of enzyme. And then we've also, at BioMarin, we evaluated the uptake of this enzyme following ICV administration. And we have these little ICV ports, and the mice are running around, and we can dose them every week. And we did dose them in this experiment weekly for 8 weeks. And we showed that the beta-gal weekly injections resulted in clearance of lysosomal storage using a histology marker, LAMP1, and also clearance of neuropathology markers, which are associated with the disease as well. So these results are really, really encouraging. And so what we're doing at CureGM1, because the program got terminated and we really want to get this into patients, we want to have something there for them. CureGM1 is trying to develop their own ERT program. And so far we have made a cell line and we're in the process of characterizing it, and we're still generating it actually, so we've still got a way to go. But we're at step 1. And because drug development is so expensive and normally companies do it, the foundation is trying to raise money. So we're up to step 5 now, where we're trying to generate donations, we're applying for grants, and we're looking for partnerships to try and move this forward. And this is my acknowledgement slide. Just a big thank you to our amazing leader, Christine. Not only is she a super mum, but she's also a great project team leader scientifically, and everyone on the ICV-ERT team at Biomarin and the ERT program in Adelaide, and a special shout out to some Ultragenics folks that have been helping us with this, Emil Kakis and Dave Jacoby. Thank you.